This video is a demo of what we would actually do in person for an osmosis lab. It starts out, and I'm showing that is called dialysis tubing. Dialysis tubing is very similar to a cell membrane because it has a semi-permeable membrane and it lets water move in and out of it. So if we look back at it, you can see that it is tied to each end, and then it put a specific solution inside of it. So now what you're going to do in your lab is it's going to have you take a graduated cylinder, and you have I think on your lab you have 5 different types of sugar solutions. So on the first one, A, you're going to fill it up. Now this is going to be your control group, so you're not going to add any sugar into it. B, you're also going to fill it up. I chose to do 500 milliliters. This also is a 0% sugar solution. In C, we're going to do a 10% sugar solution. So once again, one of the constants here is we're always filling it up to 500 milliliters. So when you have to figure out the sugar, you put it on the scale. Now you have to get that even. I put a petri dish on there. You push the 0 or the tare, and then you're going to put your sugar on there. Now to get it at 10%, you need 50 grams. So in 500 milliliters of water, if you had 50 grams of sugar, that's going to be the mass divided by the volume, you get a 10% sugar or sucrose solution. So I went a little bit above, get that out of there. So once you have 50, I'm going to add it to C. Use a stirring rod to make sure I get that completely dissolved. Once that is dissolved, we're going to go to D, and this one I changed it. It's going to be a 20% solution. So once again, I fill it up to 500. Since it's a 20% sucrose solution, you have to tare that back to 0 again. And we're going to add 100 grams of sucrose. Now since my petri dish isn't large enough, I'm going to do 250 grams and put them both in there. I'm going to fast forward through that really quick. And then the second 50. Okay, then stir in the sucrose. So now you have a control group is your A, B with a 0% solution, C with the 10% solution, and D with the 20% solution. Now, you have to weigh the cells. The idea here is that each of these cells have a specific solution. The dialysis tubing that I'm going to put in tube A is a 0% solution. Like I said, A is our control, so everything has to be just normal. And we'll see which way water will move in and out. B, C, and D all have a 10% sucrose solution. So we are going to weigh each of these dialysis tubing bags. The idea is to see if it gains weight, which would mean that water moved into the dialysis tubing, or if it lost weight because the water moved out of the dialysis tubing. So we will weigh each one. There's A, so I put it on the scale. Now each of these can have different weights. It just depends on how much solution was put in there. It may not have been completely even. Now we grab B. B has a 10% solution, so it does have some sucrose inside of the dialysis bag. And you would record that on your data table as the original, and then it's put into the B test beaker. There is C, which is also a 10% solution. And last one is D, which I think was filled up a little bit more. It was just that I filled it up too much. It doesn't have anything to do with the amount of sucrose. So now I've put all of my cells in the beakers. Now I wrote on the board, here's what it kind of looks like. A is our control. So there's a 0% solution outside of the cell, and there is a 0% solution inside of the cell. B is a 0% outside of the cell, but there is sucrose inside of the cell. C, you have both a 10% inside and outside, and D, you have a 20% solution and a 10% solution inside. Remember, the higher the number, the higher the concentration. Okay, now on your lab today, you're going to have to identify the different types of solutions. So the first, isotonic. Isotonic is one that the concentration is the same on both sides. Now remember, isotonic, water is still moving in and out, but they're just moving equally. Now if it is in a hypertonic, the concentration of the solution is higher inside of the cell. So that would mean it has more of the sugar solution inside. And water is going to move out to try to reach equilibrium. In this instance, the cell is going to shrink because it is losing water. The hypotonic solution, the concentration of the solution is lower inside of the cell, and water is going to move in to the cell, causing it to get larger and possibly burst. Now remember some of these. All the things that I talked about today on this lecture will help you with your lab. I'll also go through your lab and kind of explain that